Thanks to Anton and Gelta for this invitation to speak here today. I'm really excited to tell you about some of the work that we're doing. So my lab is really interested in trying to understand how the mammalian cortex performs the complex computations that are necessary for cognitive processes like decision-making, short-term memory, and flexible sensory motor mapping. What I want to tell you about today is one project where we've tried to uncover some of the functional organization of parts of cortex for navigation decisions in dynamic environments. This is the work of two really talented graduate students in the lab, Si Zhang and Salman Shta. And so this is a really kind of broad project with many different aspects to it. And I'm just going to tell you about one slice of it that really, I think, connects well to some of the topics that we're discussing in today's workshop. So as a lab, we're really interested in understanding learned and flexible navigation decisions because we think these are fundamental aspects of natural behavior. So suppose you're walking down the street and you reach an upcoming intersection and you have to decide whether to turn to the left or the right. You might have something like your favorite restaurant in mind that you're trying to reach. And then you might, over time, realize that you can use things like certain landmarks or street signs in order to guide your decision of whether to turn to the left or the right. But on other days, you might have other internal goals in mind. For example, you might want to be going, rather than to your favorite restaurant, you might choose to go to one of the local fruit markets, and then you might want to actually turn left at this upcoming intersection. So we think that these kind of learned and flexible navigation decisions are really important in our daily lives, but they're also important in the daily lives of the subjects that we study, of mice. And furthermore, we think they're really a nice tractable system to study cognition in a laboratory setting. So now navigation decisions require a few different features that we think are really quite exciting and interesting to think about for cognitive processes. And these include the ideas of continuous feedback and the ideas of integrating multiple features. So it might be tempting to think about these types of complex processes in a very kind of linear feed-forward manner. So you might want to think about this as perhaps sensory stimuli being used to drive navigational goals or decision-making, which can then be used to implement motor outputs like forming navigational trajectories. But we know that it's more complex than that. We know that there are feedback loops, and I'll just show you two examples here. For example, there can be fast feedback loops for things like sensory motor loops. And furthermore, there can be longer feedback loops such as for using outcomes to evaluate whether goals or decisions were valuable or not, and update future goals and decisions. Furthermore, we think that each of these processes really does not stand on its own, but requires the integration of many different features. For example, it's not just sensory processing, but rather sensory processing must be incorporated in the context of a constant ongoing movement. Furthermore, decision-making requires incorporating things like sensory information, positional information, movement information, information about past outcomes. And furthermore, things like navigational trajectories really have to take into account things of like, what is your goal or your decision and where is your position? And so we wanted to understand some of the functional organization of cortex that could kind of incorporate all these different types of processes. Now we know that because all these processes are involved, navigation decisions likely involve a wide network of cortical and subcortical areas. Subcortical areas are undoubtedly really important for these types of behaviors, but we have not studied those yet. So I'm just going to focus for today on the idea of cortex. And just to give you an idea of some of the things that have been found that provide some of the foundation. For example, we and others have studied posterior parietal cortex and identified that this area can be important for forming things like navigational sequences or generating upcoming choices for movements or posture. Areas like visual cortex are obviously important for perception and have recently been shown to be important also for having spatial information. Higher visual areas, for example, can be important for complex visual processing, such as for understanding landmarks that can be important for navigation. Retrosplenial cortex has been widely involved in navigation studies for things like place cells and head direction cells, or for combining ego and allocentric reference frames. And then, of course, frontal cortex has been thought to be important for things like planning and reporting actions. So this gives us kind of a flavor of this wider network. And so we wanted to understand some of the functional organization. So the goal for today's talk is to discuss some of our findings about the functional organization of cortex underlying flexible decisions. And so when one starts to think about all these different cortical areas and starts to try to assign functions to these different areas and studies them kind of one by one, trying to uncover what each area does, one starts to come to the idea that there might be a modular or hierarchical organization to these types of areas. However, recent work has really challenged some of this idea. Many studies actually recently have started to illustrate the idea that there can be highly distributed representations. Here I'm just showing you one example from the Dysoroth lab, where they used calcium imaging to look at many different neurons in many different brain areas, and they could find many cells across many different brain areas that had very similar activity patterns and a simple goal-directed behavior. So we wanted to understand if there's a way to where we could actually start to reconcile these ideas of maybe modular organization and distributed representations, or moreover, just come to a kind of more holistic understanding of what the functional organization of cortex is during such a complex behavioral task. So a few years back now, we developed ways in which we could begin to do this. We developed a virtual reality system in which we can begin to study navigation-based decisions. So the idea here is that a head-restrained mouse runs on top of a spherical treadmill. This is a styrofoam ball, and the mouse is surrounded by a large visual virtual reality environment, so the mouse can run and steer and navigate through these environments. Here's a movie of a mouse running down a virtual linear track. So what we like about the VR environments is that we have great control over the sensory stimuli that are given to the mouse. We can really nicely record the motor outputs of the mouse, and importantly, because the mouse's head is fixed, we can use kind of all the tools that are available to probe the neural circuits, all the tools that are available in the mouse. And so we developed a behavioral task for mice, which we call a flexible decision-making task during virtual navigation. The idea is that we had mice run down a Y-shaped virtual reality maze, and at the beginning of the maze, we showed them either a black cue or a white cue on a given trial, and then the black cue would indicate that the mouse had to turn to the left in order to receive a reward, whereas the white cue indicated the mouse had to turn to the right to receive a reward. Now we played a trick on the mouse where after about 100 trials within a session, we actually switched this association. So now in a separate rule, which we call rule B, the white cue now means turn left and the black cue now means turn right. So after every 100 trials or so, we would switch this association, and the mice would, without telling the mice would have to actually learn that this switch occurred through trial and error. So this is what it looks like within a trial. A mouse runs down a Y-shaped maze, sees a black or white cue, has to turn left or right, then enters a feedback and trial interval period where it gets feedback about its correctness and or reward. And so what's important is that as the mouse runs down this maze, it has to incorporate information, sensory information about the cue that it sees, and internal information about its belief of the rule in order to update it, in order to choose an action, turn left or right. And then at the end of the trial, the mouse has to then incorporate what cue it saw, what choice I made, and whether it got the trial correct in order to update its understanding of whether it's in rule A or rule B. So this is what an example behavior looks like for a mouse on a given session. So it can start doing very well, about 100% correct on rule A. Then without telling the mouse, we switch to rule B. The mouse's performance actually drops below chance. It's still doing rule A, and then through trial and error, it learns that it's in rule B, starts doing rule B quite well. We do another switch and then recovers and starts doing rule A again. So this is what it looks like for switch to line behavior, where you can see that the mouse can do quite well, and then over tens of trials can recover after a switch has been made. So now we wanted to understand during this task, what is the organization of some of cortical activity, specifically in posterior cortex, for areas that we know are important for performing this behavioral task. So we thought we could image individual brain areas one at a time, as has been traditionally been done in studies, but we thought this might give biased views of and not allow comparisons across different areas. So rather, we thought of sampling neural activity across posterior cortex without respect to area boundaries. So the idea is that we, on a given day, we went to a single field of view and did 2-photon calcium imaging volumetrically. We could then take an image of the blood vessels, zoom out to the full cortical window and align these images to the vasculature pattern. We could then align every single cell that we imaged within that field of view. Then on different days, we could actually image many different fields of view across many different parts of cortical space. We could then do wide field retinotopic mapping. So that with retinotopy, we could identify different brain regions such as V1 or this area AM. We could then align this retinotopic mapping to the retinotopic mapping from the Allen Institute Common Coordinate Framework, so that we could actually start to place every single neuron that we imaged into a common reference frame, even across mice, into this Allen Common Coordinate Framework. And furthermore, we could then, even if we wanted to, segment these into different brain areas. For example, we could look at things like retrosplenial cortex, or we could look at things like neurons in V1 or in this brown area. This is area A. This is like an anterior part of PPC, or other areas like AM, PM, and MM. So we collected a large data set We imaged hundreds of layer 2, 3 cells simultaneously on each day. And we collected about 300,000 neurons across 300 sessions. And so we think this is a really kind of unprecedented size of a data set for a single task and a single decision-making study. So we could start by looking at the activity patterns within individual neurons, and we observed diverse activity patterns. I'll show you 3 example neurons here. So here I'm showing you each row is the activity of a single neuron on a given trial. This is position along the maze here, and this is time within the intertrial interval. Here you can see black Q trials where the mouse turned left, black Q trials where the mouse turned right, white Q trials where the mouse turned left, and so on. And you can see that this cell was active and selective for when the mouse saw the black Q instead of the white Q. We can now look at a second neuron. We can see that this neuron is active only on one trial type when the mouse saw the white Q and turned left, and only at one position within the maze. So it had conjunctive activity for the position in the maze, for the Q, and for the choice. And then we could look at a third neuron type, and this neuron was really active kind of on all different trial types, really in the intertrial interval. So it's nice to look at these individual examples, but really with so many neurons, we needed a way in which we could come up with systematic ideas to uncover the encoding that's present within these individual neurons. So we therefore turned to developing a generalized linear model in which we can quantify the encoding profile of single neurons for kind of all of the task and behavioral variables that we've measured. So the idea, and I'm not going to go into the details here, but very big picture is that we take the activity of a single neuron and we try to fit it and predict it based on all the variables that we can measure or define within the task. So we have the task variables, things like the cue that we show to the mouse, the choice the mouse makes, things from previous trials. Don't have time to tell you about this, but I've done a lot of behavior modeling where I try to identify the decision-making strategy of the mouse on every trial. And we also have things like the instantaneous movements of the mouse, such as its running speed and acceleration on the spherical treadmill. And so we can feed all of these into this generalized linear model and try to predict the activity of an individual neuron. So this is what it looks like for those 3 example neurons that I showed you. We have in black the actual data from the neuron, and in red the prediction, and we can quantify how well the model is performing using a metric called deviance explained. This is conceptually similar to the amount of explainable variance. And then furthermore, we can then leave out one of these different features and ask how that affects the performance of the model. So we can come up with what we call an encoding profile, where we can say how much, say, the cue or the previous outcome affects the performance of the model, and thus how much that affects the encoding of the neuron. So this is really exciting to us because now we have the position of every neuron that we've imaged, and we have an encoding profile for every neuron that we've imaged. So we can then go and start to make maps to see if we can understand something about the functional organization across posterior cortex. So we can start by looking at one class of variables, such as the instantaneous movement of the mouse, like the running velocity of the mouse. And so what I'm showing you here is a scatter where each dot is an individual neuron. This is V1 to orient you. This is retrosplenial cortex, and this is posterior parietal cortex. And we can color code each neuron by its fraction explained deviance due to that variable, like the movement. So higher values mean that that neuron is more strongly encoding, or that neuron is more strongly related to that neuron's activity. And so what you can see by looking at this map is that we see very strong encoding of movement in this anterior part of posterior parietal cortex, but we see cells that have strong movement encoding kind of spread throughout cortical space. And furthermore, almost all the neurons have kind of non-zero movement encoding. We can see this maybe a little bit clearer if we smooth this map so that you can clearly see this gradient of strong movement encoding in this anterior space that kind of fades off and as one goes more in the posterior direction. So this is interesting to look at this map for movement, but we can now do this for other variables as well. So what about if we look at the visual cue and look at the encoding of the visual cue? As you might expect, we see very strong encoding of the cue in primary visual cortex. This is also present in some of these secondary visual areas. But once again, you can see cells with strong encoding of the cue that are kind of sprinkled throughout in many different other areas of the cortical space. So these are 2 parameters that seem to have different gradients, whereas movement was high up here and faded going posterior. Cue is high in V1 and fades going more anterior. And so we thought, what is a third parameter we could look at? What about the choice of the mouse? Now we're going to have a brief divergence here, because when we started to think about choice, this got quite interesting to us, because we thought we could talk about the choice of the mouse just as whether the mouse turns to the left or the right. But we realized as the mouse is running down the maze, it might actually form its choice at different time points in the maze on different trials. So we thought that perhaps we could understand the time course of choice formation by actually looking at the running patterns of the mouse. So just to remind you, because the mouse is on the spherical treadmill, we can actually record the rotations of the spherical treadmill, the velocities of the spherical treadmill about 3 different axes. We can look at, for example, one of these axes, like the roll axis, that controls whether the mouse moves to the left or the right. And as a function of position along the maze, we actually saw that when the mouse chose to go to the left in blue, it actually started to run more to the left early in the maze. And when the mouse made a right choice, it actually started to run more to the right. However, there was a lot of variability between trials. For example, on this trial 1 here, the mouse turned to the left very early, whereas in this trial 4, the mouse actually turned to the right first and then moved back and turned to the left later on. But on average, mice tended to run to the left when they were making a left choice and tended to run to the right when they were making a right choice. So we thought we could use a decoder. Here we used a recurrent neural network to see if we could actually predict which choice the mouse was going to make based on its running trajectory, and indeed we could. I'm not going to go through the details of this, but the output that we get from this is essentially what we call a dynamic choice signal. This is the probability at any moment that the mouse is going, that we think the mouse is going to turn left. So here we can see that on these left choice trials, this probability kind of grows on individual trials as a function of position in the maze, but we once again see lots of variability across different trials. So there's lots of checks that we've done on this to make sure that this seems like kind of a choice related signal. I'm happy to discuss those later, but in brief, we think that this is a behavioral estimate of kind of choice formation and kind of gives us the time course of choice formation. And interestingly, this is much better related to the neural activity for choice that we observed than just the binary variable of whether the mouse ultimately chose left or right. Okay, so now what does the spatial map look like for the choice encoding? We found that choice was also highly distributed, where we could find cells that were high for choice encoding throughout many different parts of posterior cortex, but they had the highest density and the strongest encoding was present in retrosplenial cortex, and this kind of faded going from medial to lateral, even going into posterior parietal cortex. So choice encoding was also highly distributed, but was strongest in retrosplenial cortex. I note that its magnitude was a little bit lower, which we think is probably makes sense because it's an internal variable as opposed to like a visual cue, which is something the experimenter controls. And I'll note that in retrosplenial cortex, for example, cue encoding and choice encoding were about equal in magnitude. So we thought this was quite exciting because we think we had found 3 prominent spatial gradients of encoding in posterior cortex. We saw this cue encoding, this movement encoding, and this dynamic choice encoding. And really they originated from different areas, from V1, this anterior part of posterior parietal cortex, or retrosplenial cortex, and kind of faded off in different directions. So we thought that this could be kind of dominant features of the organization of posterior cortex, but we wanted to investigate this in a little bit more of an unbiased way because here obviously we're choosing out these features that we thought were quite important. So what we did was we tried to test the idea that if there were these functional specializations for given areas, then there should be unique encoding profiles for the cells that are present within those areas. So we reasoned that we should then actually be able to decode where a cell is located in posterior cortex, which area it's located in, depending solely on its encoding profile. So if we take its encoding profile, such as how much each of these variables contributes to the activity of a given neuron, we could then go to different spots in cortical space and use a logistic decoder to estimate the probability that we would find that a neuron with that encoding profile would reside at that spot. So we could do this, for example, if we took neurons that were in V1, and we could see that the probability that we would find them in V1 was much higher than we would find them in, let's say, retrosplenial cortex or other areas. This was similarly true for trying to decode the cells in area A or cells in RSC, where it seems like we could very well decode the actual position of where those cells were residing. And we think this then indicated very nicely that there were then kind of specialized activity patterns in these different areas that would inform us of their cortical location. This was interestingly not quite the case for these kind of intermediate areas like PM, AM, and MM, and this makes sense because this is kind of where we think kind of all of these 3 gradients come together and start to intermix. Now, what gets even more interesting to me is that we could then do an unsupervised factorization of these encoding profiles and see if we could uncover these same 3 spatial gradients. So what we did was we took all these neurons we had and we took these decoder locations and all the decoder values, and we could do non-negative matrix factorization and found that actually 3 factors very well explained these encoding profiles across space. And when we looked at the weights for these for these factors, we could find one factor that seemed to very well match up with our visual type of gradient, one factor that really well matched up with our choice type of gradient starting in RSC, and another factor that seemed to very well match up with our movement type of case. So we thought that using kind of this unsupervised factorization, we could start to uncover these three different spatial gradients that were present, indicating that this seems to be really prominent features of the organism.
10 minutes, Keith?
Okay, thank you. Really prominent features of the functional organization of cortex. But this isn't the whole story. There's a lot more going on that I think is really critical to point out. So as I've tried to mention throughout, these representations were also very highly distributed. So these are the scatters that I showed you for cue, movement, and choice. And once again, you can see that pretty much any area you go to will have encoding for each of these different variables. And furthermore, each variable will be encoded in multiple different spots. And furthermore, each area will encode many different features. So that even though there can be functional specializations, these representations tend to be highly distributed. Furthermore, we're only looking at individual variables here like cue, movement, or choice one by one. But when we looked at the representations in single cells, we found that cells often had conjunctive type of coding for multiple task and behavioral features. I'll just show two examples of that. For example, if we zoom on retrosplenial cortex and we look at the joint distribution of encoding for something like movement and things like task variables, we could look for cells that say had low movement, or cells that had high movement encoding, or cells that had low task variable encoding or high task variable encoding. We actually found that the majority of retrosplenial cortex cells fell in this middle area that had high movement and task conjunctive encoding. Furthermore, if we did something similar where we looked at the encoding of cue and the encoding of choice, in retrosplenial cortex, we actually found a high density of cells that had the encoding of both of these types of variables together. So therefore, cells were not just encoding one variable, but actually had this conjunction. And so we wanted to quantify this in a little bit more detail and come up with a quantitative comparison of encoding across cortical areas. So what we did was we developed an encoding space for all of our imaged neurons. So we could come back, and I'm not going to go through all the details here, but in broad terms, what we did is we could go back to our decoders of trying to identify a cell's location, and we could just do a simple PCA to come up with a linear embedding. So what we got out is this two-dimensional plot where each dot is an individual neuron, and what this embedding is doing is putting together cells that have very similar encoding profiles very close to each other, and cells that have very dissimilar encoding profiles very far from each other. And so we could just color code each cell based on how strongly it was weighted for, let's say, factor 1, the visual factor, and we could see that kind of occupied one part of this triangle, or for a second factor like choice, that occupied another part of this triangle, or movement occupied a third part of this triangle. And what was then quite exciting is we could then go and understand where cells that belonged to the different brain regions actually reside within this encoding space. And so we could look at, for example, the contour for all the cells that were in V1, shown here in blue. And you could see kind of that there was a little lobe that stuck out that corresponded to high visual encoding. We could see for area A, this in brown, this area that kind of stuck out for high movement encoding. And in purple, retrosplenial cortex stuck out for this kind of high choice encoding. But what was really striking was how much there was overlap between the encoding between all these different areas. So this extensive overlap in encoding really indicated that there were these distributed representations. Furthermore, the highest density of cells actually fell in the center here, not at the extremes between kind of these pure selectivities, and this kind of being at the center kind of indicated that there was extensive conjunctive coding that was present. So we think that altogether, these start to reveal three key features of the functional organization of posterior cortex during these navigation decisions. We found these functional specializations that show up as these spatial gradients across cortical space. We found these distributed representations where each feature is encoded in many different areas in a distributed manner. And we found that single cells actually integrate and have conjunctive coding. And we think all this comes together really nicely because it fits with what we think is actually needed for these navigation decision tasks, where you can kind of have this linear feed forward type of idea of computation that can fit with functional specialization, but with feedback, that means that things have to necessarily be distributed. And ultimately, these features must be integrated in order to perform the computations that are necessary for navigation. So going forward, we think this presents really a nice opportunity to take these experimental data and start to apply them to the idea of computational models. And so here I'm just showing you kind of a schematic or a cartoon model a working model for flexible navigation decisions based on a principle that we're thinking of as integrative specialization. Here I'm just showing you 3 cartoon areas, V1, retrosplenial cortex, and area A. And so the first principle that we would come across is this idea of distributedness. This is the idea that areas actually share similar sensory, cognitive, and motor information, so that each of these areas would encode visual information, choice information, and movement information. The second principle is this idea of integrative specialization. This is the idea that areas might integrate and combine different features in different ratios. So area A might be more important for movement, retrosplenial cortex might be more important for choice, and V1 might be more important for visual information. And this leads to some predictions about the computations that are present in these areas, the hypotheses about the functions for each of these areas. So perhaps V1 is more important for visual processing, retrosplenial cortex more important for navigation decisions and goals, and area A more for movement biasing for forming navigational trajectories. And so we think that we need to bring kind of all of these 3 principles together to create models in which we can start to understand the computations within these areas, but more importantly, how these areas communicate with one another to create these distributed and integrative features that are also really prominent and dominant features of what's going on in the network. And we think that the scale of the data that we're collecting and that we're even now expanding towards will be kind of a nice starting point for building kind of multi-area computational models at finer scale than people have thought about before. And so I want to end with thanking the people in the lab in particular. I want to thank Si Zhang and Salman Shte, who did the bulk of this work. And I want to thank everyone else in the lab who's participated in lots of different ways. And to just mention, it's a great group of people to work with. And I feel very, very lucky to have the chance to work with all of them. And thank you again. And I'm happy to answer any questions.
Great, thanks, Chris. Yeah, we have some questions here. One is from Aritra Bhattacharya. Hi, Chris. How is natural variability amongst different mice? Any factor shows greater variability relative to others? Also, would there be some differences by strains?
Yeah, these are good questions. We definitely see we definitely see variability in the behavior of mice, but overall, I would say that you know, these general patterns that we observe, you know, averaged across mice tend to show up in individual mice. So we don't think that these are kind of like, there's going to be tremendous mouse-to-mouse variability in some of these overall patterns, but we have seen in other work that I'm not talking about today, that things like the training history of mice, if they have experienced other tasks in the past, can dramatically affect, for example, the implementation of which cortical areas are necessary and so on. So we think those can have a big impact, but still kind of a work in progress to figure all that out.
There's one from Shanshan Kin. Great talk. Does individual neurons shift its coding over time? So that a neuron first encodes for visual cue and then shift to encode movement?
Yeah, that's a good question. We haven't studied that here. In other projects, we've tracked the same neurons across time, and we found something that was surprising to us, which was that the representations, even after the mouse had become an expert at a task and was performing the same task every day, the representations seemed to kind of drift and reorganize across days. But the primary mode of the reorganization was that cells would kind of drop out or come into the representation rather than shifting what they were coding for. So it was pretty unlikely that a cell would move from encoding like the visual cue to the movement, and it was more likely that a cell would kind of become less active or become more active. But there was a lot of shift over days that we think is quite important, and actually ties into some of the nice kind of homeostatic type of ideas that Keith was talking about.
That's one from Terry Sinofsky. How is the hippocampus involved in this task?
That's a great question. We don't know. That's something we want to investigate very soon. Obviously, it's going to be heavily involved. It's heavily interconnected with some of the areas that I've talked about. We just haven't looked yet, but definitely something on our short to-do list.
Yeah.